Good morning, my dear students. Welcome to my channel Patel Science Academy. You are watching here Patel Science Academy, and this is your Shahzad Yunus Patel, sir. Dear students, as you know, the chapter I have started is the molecular basis of inheritance, and the topic you are going to know in this lecture is Griffith experiment, which is very interesting experiment regarding the DNA discovery. Dear students, but before we go in the depth of that Griffith experiment, you should know about the basic of gene. What is gene? So dear students, the gene means, to know about the gene, I think we must go from the basic. The body of the all living organism is made up of cell. This is the structure of cell. This is its outermost membrane, that is known as plasma membrane, and this is it consists of nucleus. So this is nucleus. So a cell, eukaryotic cell, it consists of plasma membrane and the nucleus, and inside the nucleus you can observe here this structure, colorful structure. Inside the nucleus, this colorful structure is there, and this colorful structure is called chromosomes. And this chromosomes, on this chromosomes there is a double stranded structure, genetic material, DNA is present, and the small fragment of the DNA is called gene. So what is gene? The small fragment of the DNA. What is DNA? The genetic material, double stranded material, which is present on the chromosomes. What is chromosome? The genetic material of the cell present inside the nucleus. What is nucleus? Nucleus is the structure present inside the cell. And what is cell? The basic unit of the living organisms. All these cells together forms a tissue, means the group of the cell is called tissue, and that tissue together makes different organs of the body, like the heart, brain, etc. And that organ is responsible for performing the different processes, and that processes together is called a system. And there are different system in our body, like the circulatory system, respiratory system, excretory system, digestive system. All that system together in the body makes a normal body. So in the body there are different systems, they have coordination and communication together. So that system is made up of different processes, and that processes is managed or run by the organs, because organs perform the different processes. And organ is made up of tissue is the group of the cell, and cell is having this structure. Inside the cell there is a nucleus, inside the nucleus there is a chromosome, and on the chromosome there is a DNA, and DNA consists of small fragment, it is known as gene. So this is about that gene, which is the small fragment of DNA. I explain it because we have some concern related this DNA and this gene with this Griffith experiments. Dear students, now we'll see the Griffith experiment. What is that Griffith experiment, dear students? Before we go in the detail of this Griffith experiment, I would like to explain this slide here. Griffith in 1928, the full form of the Griffith is the full name of the Griffith is Frederick Griffith. He was a British medical officer, and he performed an experiment on the bacteria. What is the name of that bacteria? Streptococcus pneumoniae, or Streptococcus pneumoniae. So this is the name of that bacteria, Streptococcus pneumoniae. This is the bacteria Streptococcus pneumoniae, which causes the disease pneumonia in human and other mammals. Dear students, Griffith used two strains of this bacteria, of this Streptococcus pneumoniae bacteria. There are two types and two strains that were used. That was used by the Griffith for his experiment. And why he used two strain? To find the cure of the pneumonia at that time. So dear students, which was a common disease, common cause of the disease at that time, because pneumonia in that year 1928, there was a killer disease, and there was no cure for that disease. That's why he used two strains of Streptococcus pneumoniae bacteria at that time to find out the cure of that disease. And that, what was the two strains? Strain number one was S strain of bacteria, and strain number 2 was R type of bacteria. Both strain, this is S strain and R strain, both this strain was of Streptococcus pneumoniae. Let's see the some detail of the S type bacteria, Streptococcus pneumoniae. S type bacteria, it was virulent, it was smooth, and it was pathogenic, and it was encapsulated. And about R type, R type was non-virulent, it was rough, it was non-pathogenic and non-capsulated. So you can note down this point about S type. I repeat, S type was virulent, smooth, pathogenic and capsulated. R type was non-virulent, rough, non-pathogenic and non-capsulated. So dear students, Frederick Griffith looked at the pneumonia bacteria trying to figure out what made people die. So he used these two steps, these two strains of this bacteria, as I told you, S type, which was virulent, smooth, pathogenic, encapsulated, and R type, which was non-virulent, non-pathogenic, rough, and non-capsulated. So we'll see the detail of this both of the strains of this Streptococcus pneumoniae on the mice one by one. So dear students, let's see the detail of this Griffith experiments here. So as I told you, Griffith conducted four steps on this bacteria. Which bacteria? Streptococcus pneumoniae. And in the first step, what he did, when he injected bacteria of R strain to the mice body, the mice survived because it did not develop pneumonia. So what was that first step? First step was the injection in the body of mice, of which strain? R type strain. So this is that R type strain, which was avirulent or non-virulent, which didn't cause death, and it was non-pathogenic. So what happened when he injected the R strain bacteria into the body of the mice? Mice was surviving, there was no pneumonia disease, and because of no pneumonia, mice was normal. So this is the first step of the Griffith experiment. I am explaining here randomly all these steps because in the slide there is a random step given here. So this is first step. What happened in the first step? He used R strain of this pneumonia and injected into the body of the mice. And what happened? Mice died? No, there was no death of the mice. Mice survived because there was no pneumonia, because this R strain is non-pathogenic, avirulent or non-virulent, and because it is having rough membrane, and it is non-capsulated. Now we'll see the second step. What happened with the second step? In the second step, dear students, when he injected the bacteria of S strain, means now there's a turn of S type strain of this Streptococcus pneumoniae to the body of the mice. What happened to the mice? The mice developed pneumonia and died. So this was that second step, which is given at first number. What was that second step? Let's see the second step. This S strain of the Streptococcus pneumoniae, it has been injected into the body of this mice, and because of this S strain, which was pathogenic, which was virulent, which was killer, and which causes the disease, because it is pathogenic in nature, it is having smooth layer or membrane, and it is capsulated. So when it is injected into the body of this mice develop pneumonia and pneumonia disease, and because of the pneumonia this mice died. So this was second step. What we have concluded till now? Till now we have seen, if R strain is injected into the body of the mice, there is no pneumonia, mice survive. Second step, if S strain is injected into the body of the mice, what happened? Mice died because this S strain is dangerous. It is pathogenic, virulent, and capsulated, and having smooth membrane, and it causes the death. So till now I hope you have understood two steps. Now we'll see this step number third. What happened in the step number third? So dear students, I hope you have learned about the first step. First step, R strain is injected into the body of the mice, and R strain is not responsible for the death of that mice. And then the second step, S strain is responsible for the death of the mice, because S strain is responsible for causing the disease pneumonia, and because of pneumonia mice died. In the third step, now we'll see the third step. In the third experiment or third step, what happened? Griffith injected the heat killed S strain bacteria to the mice. Now this Griffith has killed this S strain by hitting it. He heated this S strain type, and because of the heating this S strain bacteria was killed, and now this heat killed virulent S strain. Now he injected into the body of this mice. Then he observed what happened to that mice. Did he develop pneumonia? No, he didn't develop pneumonia disease, and heat killed S strain bacteria. It once again the mice survive because there is no pneumonia. But which thing is responsible here for the death of the this mice till this step number third? Only one thing, that is the living S strain. Living S strain is responsible for the death of the mice, as we have seen till now. Now we'll go for the last step of the experiment of this Griffith. So what is this last step? In the last step, he injected the mixture of heat killed S bacteria with the live R strain of the bacteria to the mice body, and then he observed the mice died because of the development of the pneumonia. So dear student, you may have confusion here in the fourth step. This is that fourth step. What happened in the fourth step? He used the mixture of these two strain. One strain was a virulent, that is R strain, and the another strain was S strain. But this S strain this time was heat killed, means it has killed by the heating. So as we have seen in the step number third, this heat killed S bacteria is not responsible for the death of this mice, and R strain in the first step we have seen, it is also not responsible for the death of the mice. So he was considering that the R and the heat killed S strain, both of this strain, if I am mixing this two strain with each other, so it will not cause the death of the mice, but the conclusion or the result was mice died. So he was surprised why mice died, because of the pneumonia. So he was very surprised, because R strain in the first step is not responsible for the death of the mice, and heat killed S strain in the third step is again not responsible for the mice, but the mixture of this heat killed S strain and R strain, living R strain, both of these when they get mixed with each other, so it is responsible for the development of the disease pneumonia. So what was the conclusion of this Griffith experiment? Now we'll see the conclusion of Griffith experiment. He concluded that The conclusion was, according to the Griffith, there was a transforming principle. What was that transforming principle? Griffith actually Streptococcus pneumoniae bacteria, as I told you, was working to find out the cure of the pneumonia. The harmless live bacteria that is having rough skin, it mixed with the heat killed pathogenic bacteria in the fourth number step, and it causes the fatal disease in the mice, and because of that mice died. So he concluded that there was a substance he concluded that there was a substance passed from the dead bacteria, means that heat killed S bacteria, to the live bacteria. Which was live bacteria? R strain. What was dead bacteria? S, heat killed S strain. So this heat killed S strain, according to the Griffith, has passed some materials of substances into this R strain bacteria, and because of that substance which has been passed from this heat killed S bacteria into this R strain of the Streptococcus pneumoniae, because of that substance, this R strain become virulent and it causes the disease pneumonia, and because of the pneumonia the mice died. So question was, what was that substance which has been transferred from this heat killed S bacteria into R bacteria, and because of receiving that substance, R bacteria become S bacteria and causes the death. By the Griffith, that term given to that substance was transforming principle, and actually nowadays that transforming principle is DNA. But Griffith couldn't give this name DNA, deoxyribonucleic acid, to that substance. He just used transforming principle to that substance. So this was Griffith experiment, in which the conclusion is DNA is responsible for the death of that mice, but he called that DNA transforming principle. This is about this lecture. Thank you very much, dear students. Please do subscribe my channel, like my video, share with your friends and with all the needies who want to learn about this. And this is about the Griffith experiment from this chapter, molecular basis of inheritance. And dear students, please do press the bell icon to receive my instant notification of every new video. And please, on the YouTube, you can go in for my channel Patel Science Academy. There are so many lectures for the different topic, for the different chapters. You can learn many more things from that chapters. Thank you very much for watching my video. Stay home, stay safe. Thank you.